Dear students, Assalamu alaikum wa rahmatullahi wa barakatuhu. This is lecture number 4th pertaining to animal kingdom. In lecture number 3, we understood about the germ layers, whereby I made you understand that animals could be diploblastic or triploblastic, depending upon how many layers are present in the embryo, or whether the organism is developing from 2 germ layers or 3 germ layers. Then we started the discussion of metamerism, which we will complete in this lecture. Okay, so the 7th feature on which we can again divide animals into different types, that feature is metamerism. Now, I made you understand what metamerism is. Metamerism is the repetition of some structures in the body of an organism. So what is metamerism? It is the repetition. It is the repetition of structures in the body of an organism. The best example to understand about metamerism is that of an earthworm. So let me take, for example, annelids. When I say annelids, you have to get into your mind earthworm. Okay. So what do you see in earthworm? You see that their body is made up of certain segments, which you can see externally. Their body is made up of segments like these. Let me make over here. So you see an earthworm body, something like this. And you see these segments, a chain of these segments forming the body of an earthworm. Okay. Are you able to see them externally? Yes. So an annelid's body has external. So what is the word I can use? Repetitive segments, because you see they have the first segment followed by the second segment followed by the third segment followed by the fourth segment. So annelid's body has external segments. Okay, and you know the property of these segments. Each segment is, having a set of internal structures also. These external segments, which forms the body of an annelid, these are called, let's remember that also, they are called metamers. What do you call these structures or segments? You call them metamers. Okay, even you will see that the body is internally also segmented. Okay, externally you see them that they are segments, and you call each segment a metamer. So this is a metamer, this is another metamer. Internally also these metamers are segmented. So you will have internal separations also between one metamer and another metamer, and those internal separations or internal walls which will separate one metamer from another metamer, they are called septa. Okay, so one metamer is separated from another metamer because of these septas. So you see that such a condition where an organism is both externally as well as internally made up of segments, and such segments are working in coordination with one another. That means this segment is dependent on this segment and it is dependent on this segment. They do not work independently. They are dependent on each other. Such organisms are called metamerically segmented organisms. What do you call them? You call them metamerically segmented. So they are metamerically segmented. They show metamerism. Okay, they are metamerically segmented organisms or animals. That means they show metamerism. They show metamerism. They show both external segments as well as internal segments. Okay, this was the case with annelids. And you have to remember that these segments, they work togetherly. They are dependent on each other. These segments are dependent on each other. They are not independent. They are interdependent. So these metamers are what? They are interdependent. In case of what? In case of metamerically segmented bodies, or in case of true metamerism, which is shown here by annelids. Okay, you have some animals, like you can talk of your own body. We are vertebrates. So let us take another example. This was the example of annelids which show metamerism. Let us think about ourselves. Do we show metamerism? This is a very important question now. Do we show metamerism? Let me encircle it. Let us take second example, those of vertebrates. So if we talk about vertebrates, that means it involves all. It involves fishes, it involves birds, it involves your reptiles, amphibians, and mammals. That means you also. Are we metameric? Is our body made up of any repetitive structure? Is our body made up of segments? The answer is yes. But we are not externally segmented. Vertebrates are not externally segmented. In them, you will see that there is only interdependence. null metamerism. That means inside our body there is repetition of some structures. Now the question which can come in your mind is that what are those structures which we will find inside our body, that too in a repetitive manner? So what type of repetition, what type of repetition do we see? We see the repetition of vertebras. We see the repetition of vertebras, the repetition of vertebrae. The bones which make your vertebral column, they are present in repetition. So that means internally our body also has a sort of metamerism, a sort of repetition of some structures, like the repetition of your bones of vertebral column, which you call vertebrae. Similarly, we have repetition of ribs. Okay, there are so many ribs present in our body and they are present in a repetitive manner, a sort of repetition, which is resembling somewhere the structures which were called metamers in annelids. So we don't have metamers as such, but there are some internal structures which are present in a repetitive manner, like our vertebrae, like our ribs, like our muscles. They are also present in a repetitive manner. So this is the way by which we can justify that our body also shows metamerism. But only which metamerism? Internal metamerism. Let me give you one more example. Let us talk about the arthropods. Okay, the third example is our arthropods. Let me take a different color. Let us take the third example, those of the arthropods. Why isn't it writing? Or arthropods. Arthropods involve your insects, say for example. Okay, arthropods are very peculiar. See, in them you will see if you look at an insect, if you look at some flies, or you look at honeybee, you will find that their body is externally segmented. So they show external external they show external segmentation. They show external segmentation. Their body shows external segmentation. Externally, ahead inshallah we'll be saying that arthropods body, we will take into consideration different examples whereby I'll show you that their body has external segments, what we call prothoracic, mesothoracic, metathoracic, and even their abdomen is divided into segments. Okay, we'll see 10 abdominal segments present in the arthropods. So we are here able to conclude that arthropods show what? External segmentation. The body of an insect will be having a prothoracic, a mesothoracic, and a metathoracic. We will have a segmentation like this present in the arthropods. Okay, but if we look at the internal structure, internally in arthropods, there will be no segmentation. Internally, there will be no segmentation. Let me erase first of all this was an idea I was giving you for external segmentation. In arthropods, you'll be able to see that their body has external segments, but internally there is no segmentation. That will be the case with arthropods. Although you have to remember that annelids, arthropods, vertebrates, all these higher organisms, whatever is right now on our whiteboard, so we are able to see at annelids, we are able to see at vertebrates, the higher animals, we are able to see arthropods, all of them show metamerism. That means the body of all these organisms is made up of these repetitive structures. But the difference is that annelids show internal. What is the property with annelids? Like earthworm, they show internal as well as external segmentation. Vertebrates show only internal segmentation or metamerism. Arthropods show external segmentation, but they do not show any internal segmentation. Now, there are some organisms whose body doesn't show any segmentation. Okay, you can use the word segmentation also for metamerism, because what we concluded that here we saw that the body is made up of segments. So the other term you can use for metamerism is segmentation. Then you have to understand whether it is segmented or whether it is not segmented, but metamerism is a sort of segmentation. So you will have, let me give you a note over here. Let me clear these drawings. Okay, so now you have some cases. Say for example, if we talk about flatworms, okay, if we talk about flatworms, or we talk about the roundworms. Once I use the word flatworm and roundworm, I'm talking about the platyhelminths and the nematohelminths. Flatworms and roundworms, in them the body is not segmented, so they have unsegmented body. You have to remember this. They have unsegmented body. That means no point of metamerism. They don't show any metamerism. Their body is unsegmented rather. But if you look at tapeworm, okay, tapeworm. Tapeworms are an exception. See, their body is segmented. Tapeworm's body is, you will see their body in which you will be able to see the segments. There will be segments, but these segments will be independent of each other. So there's a point to be noted here. You saw the same thing in earthworm also, but I told you over there that each segment is dependent on other segment. That means the metamers are interdependent. But in case of tapeworms, what is the point over here is that again here the body is segmented. Okay, the body is segmented. So you have a sort of segmentation is there. But this segmentation is non-metameric segmentation. So what's wrong with this? This segmentation is non-metameric segmentation. What do we mean by that? What do we mean by non-metameric segmentation now? Dear students, here it means that the segments are independent of each other. That means the body, how we'll put it down, body is segmented. It's correct, body is segmented. We agree. But each segment is independent of other. That's the difference. Body is segmented. But each segment is what? It is independent of, what's wrong with this? Of other segment. Okay. If that is the case, that you have segmentation, but the segments are independent of other segments, then you call it non-metameric segmentation, or you call that the body is pseudo-segmented. Pseudo-segmented. So tapeworms show pseudo-segmentation. They are pseudo-segmented. But on the other hand, if you look at whom? If you look at annelids, if you look at arthropods, the insects, to be simple over here, arthropods, if you look at all higher animals other than arthropods, we'll simply write down higher animals. What have we seen here? They all exhibit metameric segmentation. So my dear students, about metameric, you have to remember that this is the true segmentation, because the metamers, the segments are not independent. Each vertebra will not function independently. So it is an entire system, and the metamer is a part of the system. So the segments are dependent on each other. If that is the case, we will call that metameric segmentation. If we see that the segments are totally independent, then we'll call it pseudo segmentation. Okay, or you can call it non-metameric segmentation. And if the body is unsegmented, we'll call it an unsegmented body. There is no metamerization. Okay, this was very important to understand. So organisms could be divided into different categories, or animals could be divided into different categories depending on the metamerization, depending on the segmentation of the body. We'll have some animals which have unsegmented body. We will have some animals which have pseudo-segmented body. Pseudo-segmented is the case where there is segmentation, but each segment is independent of other. We call such segmentation non-metameric segmentation. And we'll have some animals like our annelids, arthropods, arthropods and higher animals which show metameric segmentation. Okay, in annelids, I explained to you that they show both internal as well as external metamerization. Arthropods show only external metamerization. Higher animals show only internal metamerization. Okay, so that was the difference. All of these three were the cases of metameric segmentation, because the segments or the metamers, they are not independent, they are dependent on each other. So this was the seventh criteria. Let us understand and let us take this eighth criteria into consideration. Let us take this eighth criteria, which will again help us to divide the animals into some categories. What is that eighth criteria? That criteria is the cephalization. Very, very important. Cephalization. So we have animals divided into groups based on cephalization. Now, what is cephalization? Cephalization, to be very simple, it is the head formation. Cephalon is the head. Okay, it is the head formation. Maybe you have heard in class 10th that brain is called encephalon. Cephalon is an idea of head. So you have animals divided into different groups depending on whether they show head formation or not. So once we say head, how we can put this also, we can say that we have to see whether there is the concentration of nervous system towards the anterior part of the body. I hope you remember anterior. I've told you that if I take, say for example, this fish into consideration, say for example, if I take this fish into consideration, the part of the body which has the mouth, this part is called the anterior part. Okay, similarly, if you look at your body, the head, in our head we have this mouth, and that's why this is called the anterior part of the body. The part which has the anus is the posterior part. Similarly, man, our back part, back lower part, is the posterior part. So once we say head, we are talking about our central nervous system. So we can put it in other terms also. We can say that cephalization is what? It is the concentration. What is it? Concentration. Because once we talk about head, we basically talk about neurons, we basically talk about our nervous system. So head formation can also be said as concentration of the nervous system. Concentration of the nervous system, where? Towards, where is it located? Towards anterior or posterior? Towards the anterior part of body. We have to analyze whether this is happening in animal or not. So depending on that, we can see that CNS, whether it is formed, whether head is formed in animal or not. So obviously lower animals, in lower animals will not see a proper head formation. We'll see higher animals showing this proper head formation. So I can show it, or I can make you understand this way also. Once we talk about cephalization, we can remember about it as concentration of central nervous system, because head is a part of our central nervous system. So here I said concentration of the nervous system, and where is the location? Towards the anterior part of the body. So we have to see whether there is a brain formation or not. So it's not necessary that you will have CNS in the form of brain always. In lower organisms, you will have CNS in the form of ganglion. So either you will have ganglionic form, which is just a cluster of the neurons, or you will have a proper brain. These are the two forms of your central nervous system. So we have to see whether this ganglion formation takes place, which is the term used for head over here. Okay, we have to see whether this ganglion is formed, or whether this brain in higher animals is formed or not. So we'll generally see that cephalization, it starts from, so we'll see animals from this type, flatworms. If we talk from flatworms onwards, we go to chordates, the higher level. These are the animals. That means cephalization starts from flatworms, the lower forms of the cephalization, and then as we move ahead, we go to roundworms, and then we go to annelids, arthropods, we go to mollusks, echinoderms, hemichordates, and then we go to chordates. We'll see that cephalization is shown. So from flatworms to chordates, all of them show cephali. They show what? They show cephalization. They show head formation. So if you go below flatworms, that means if we talk about what? If you talk about poriferans, okay, the sponges. If you talk about the cnidarians, what we also call Sealant Traits. Okay, there's something. If you talk about Sealant Traits, let me take a different color. Maybe there's this problem with the black color, because I observed that with some other colors, there's no problem as such. So if you take Sealant Traits, and we take ctenophores, okay, These organisms, they do not show cephalization. In them, you will not see the proper head formation. They do not show cephalization. So cephalization is going to start from flatworms, and then it's going to advance as we move towards the chordates. Now there are some other characters which we can focus. Let me understand them in the next lecture. Take care. Allah Hafiz.